Hi, thanks for joining me. I'm Tara Smith. I'm an infectious disease epidemiologist. I wrote a column for Quanta Magazine about how our existing knowledge of coronaviruses can potentially help us inform knowing and understanding what's going on with the COVID-19 pandemic. And so I'm here today on the Quanta Magazine YouTube channel to answer some additional questions that have come in. So can you explain in simple terms why COVID-19 wasn't created in the lab? I mean, first, I think it's unlikely that that happened. I know there are some really big advocates of that, but I think what some people don't realize is how often these spillovers from animals to people happen. Okay, so there's even a term for it in kind of the field. We refer to it as viral chatter. We know this happens a lot, and most of these spillovers are not documented. So it often takes something that is pretty dramatic, like right now, like this coronavirus pandemic, or like an Ebola outbreak. A lot of these spillovers are often dead ends, and they don't spread from human to human, so they may go from animal to person, but then not from person to person, so they die out. Or they can circulate maybe undetected for a long time. This is what happened with HIV. HIV actually is not a single spillover event. It didn't happen just once. It happened multiple times with related viruses. That's why they're slightly different. They came from slightly different animals, from chimpanzees to sooty mangabees to gorillas. Again, we didn't detect them initially because they don't cause symptoms as obvious maybe as Ebola. Just based on how everything works, it is much more likely that this is a natural spillover than something created in a lab. Looking at its genome sequence, it doesn't have any markers of being altered by humans. I know, and after that was kind of pointed out, the other idea came that, well, okay, but it was in the lab and then it had just gotten loose from there, which is tougher to rule out, but if so, it was not a virus that ever was recorded as being in that laboratory. It was never identified or sequenced. I can't say lab accidents never happen, but they're certainly much less common than just those natural spillovers from wildlife reservoirs or other animal reservoirs into humans. It's tough to prove a negative, but I think it's much more likely that it was a natural event than some kind of a laboratory accident. So bats host a lot of coronaviruses. Is there something we can learn from the way bats' immune systems handle the virus? And that is kind of $1 million question too. That's something that's a lot of people have wanted to know because bats harbor not only coronaviruses, but are hosts to viruses like Ebola and Marburg, Nipah and Hendra. A lot of different pathogens come from bats, and part of that is because bats just are incredibly diverse. We have so many different species of bats that it's not surprising that we get a lot of spillovers because they just harbor their own different viral groups. Those eventually can cause infections in humans. So why don't they seem to cause harm to bats? And we don't really know. Bats are really hard to work with in the laboratory. They don't really survive well in captivity. So it makes it difficult to do a lot of experimental work on them. There's also been some suggestion that just because of the way bats live, that they fly, they have to have a high metabolism so that they can expend that energy on flight. That might also play a role in their immune system. If basically they're fevered all the time, essentially they have a high body temperature, then maybe that is just kind of keeping the viruses that they have in check. But those are all really more hypotheses than anything, because again, they're just really difficult to test because we don't have a lot of good experimental data on some of the bats. How likely is it that we will discover a really powerful drug that cures COVID-19 quickly before a vaccine gets licensed for widespread use? For most of those drugs, not all, but most, that are being kind of examined for treatment for COVID-19, a lot of them are already in use for other infections or other areas. So if we could find something that is effective and it has already been licensed for something else, it's already known to be relatively safe and has already gone through all the approvals, it's much easier to take those and kind of repurpose them for treatment for COVID-19 than it is to develop something completely de novo where you have to, determine its safety profile and everything else. But from the tests we've seen so far, and of course there are many that are ongoing, so who knows what the outcome of those will be too. I haven't been particularly impressed with the results that we've seen so far. We saw the results from some of the remdesivir trials the other day, and the ones in China really didn't go anywhere. The NIH ones that were done here in the United States seem to maybe show some progress, but that's also an IV drug, which makes it just a lot more difficult to get to a larger number of people. So if we find one of those that are already approved for other infections or other conditions, then yes, I think that those could be in place before we get to a vaccine. How long will it take to develop herd immunity? When you look at the percent of the population that will need to be infected by any kind of virus to develop herd immunity, that is based on how transmissible they are in a population. You would have to have basically about 60%, 60 to 70% of the population immune in order to reach that herd immunity threshold. Okay. Now remember, herd immunity is not like a switch. It's not all or nothing. The higher number of people that you have in the population that are immune, the more difficult it makes for that virus to find a susceptible host and to spread. So, any immunity in the population is better than none, even if it's not kind of at that herd immunity threshold yet. So if we would go over the course of, say, 12 months, right, and 40% of the population would have been infected with SARS coronavirus 2, in theory, 40% of the population then would be immune. But what if that immunity wanes after a year? So you have the people who were infected at the very beginning have maybe, are maybe no longer functionally immune. So you're not really at 40% of the population anymore. You're at a lower level than that. So those are some of the questions that we just don't have answers to, and we won't have answers. We just can't have answers without following people over time. So it's only something that we will know after 1 year or 3 years, how long that immunity lasts, and if people are really protected from reinfection or not. A lot of healthcare workers are dying from COVID, statistically speaking. Could this be because of higher original exposure? That's one of the hypotheses for the reasons why some of these healthcare workers are coming down with not only illness, but in some cases really severe illness, even though they're otherwise seem to be kind of young and healthy, is that exposure to a higher load of virus, and especially a higher load of aerosolized virus. They also did see this with SARS, especially in the early days before, before it was recognized and before measures were taken to really use like N95 respirators and things like that to protect healthcare workers, that there did seem to be kind of a dose exposure there, that the more virus you were exposed to, more severe symptoms you could get. And we have a little bit of evidence of that from the early days in China also, again, when healthcare workers were exposed to this before understanding what it was, that using, again, N95 respirators and face masks and things like that to protect healthcare workers did seem to decrease their mortality as an incidence of serious infection from that as well. So we think maybe, again, that's something that will be better resolved with good animal models and animal experiments where you can directly challenge them with different doses of the virus and look at the severity of their outcomes. So that's something that I'm sure will be coming from some of these animal studies. Please check out quantamagazine.org and subscribe to the YouTube channel where they have a lot of other Q&As with other scientists. So thank you for joining, and stay safe out there. Take care of each other. Thank you.